Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm really excited to be here today with John Lepore, who's a physician, scientist, and CEO of ProFound Therapeutics, where he is leading the team with a focus on pioneering what they're calling the expanded human proteome to identify novel drugs and drug targets. If you've listened to the podcast for a little while, you've heard us speak with a number of companies that have come out of Flagship Pioneering, which is one of the world-leading venture builders behind some pretty incredible companies, including Moderna. We're going to talk a little bit about John's background beyond ProFound Therapeutics. He spent 17 years at GSK, ultimately leading R&D as senior vice president. He's worked across metabolic, cardiovascular, and almost every area of omics-driven drug development in his time there. So I'm really excited for the conversation. We've got a lot of ground to cover in the 45 minutes that we have today. So John, thank you so much for being with us.
Oh, thanks, Patrick. Absolute pleasure. And you've had a terrific group of people on, and I'm glad to be part of that and chat with you.
Yeah, and I know you worked, you mentioned before we started recording, you worked with Eric Ingelsson while you were at GSK. We had him on a previous episode. Let's maybe start with GSK, actually, because you spent a ton of your career there. I'm interested in how you transitioned from physician scientist where you're working closely to patients to actually getting into drug development and therapeutics development, where you clearly found a groove and something that you really enjoy and can have an impact on patients at a scale that maybe isn't possible in the day-to-day.
Yeah, sure. You hit all the elements, but happy to fill in some of the more personal, specific details on that. And, almost as you've prompted, I've always just been driven by figuring out how to help patients in the most important way. And from very early on, even in college, I could still remember being just fascinated about understanding pathogenesis of disease and how sort of the scientific mechanisms really go astray in disease. And, I started, as you said, in medicine. So I trained in internal medicine and in cardiology here in Boston, where I'm recording this from. And really enjoyed taking care of patients, and was a phenomenal part of my career and training, and just to have a deep understanding of how disease works and what clinical endpoints look like and what the patient experiences, and just the personal closeness to what the patients experience was very important. And I love taking care of patients, but I really was driven more by how could I affect health, not just one patient at a time, but on a very large scale. And that led me to think about, more translational biology. And when I took a detour after the clinical training in the lab, and I did a pretty extensive postdoctoral training period, both with Jeff Leiden and Mike Parmasek, and I ended up at Penn. I had a lab there for about a 6-year period. It was really interesting. I was studying the molecular basis of cardiovascular development and really trying to understand the genetic drivers of differentiation of cells and how that might relate to human disease. But for me, fantastic training, but was far from the actual translation. And I realized that so many of my friends and colleagues and mentors had moved on to industrial settings where they were doing the same kind of science, but much more directly applying it to drug discovery programs. And I thought that was really cool. Perry Neeson, who maybe he'll happen to listen to this, I'll send him a link, was the person who talked me into going to GSK and recruited me there. He was visionary for not only for GSK, but for me personally, where I could thrive in an environment where I can start to do this kind of translational work. And as you said, I hit a group there. I really had a fabulous run. Ran several different organizations from sort of discovery all the way through development, inclusive of many of the platforms, including human genetics, and really saw how it all fits together and really helped shape a strategy and a portfolio. But at the same time, we all get calls, ignore most of them, cordially chat with a small number. But this particular opportunity to go to Flagship and to work at ProFound Therapeutics, it really caught my attention because, as I'm sure we'll describe at some point in the call, it opened up like a whole new field, an opportunity to step into something that was nascent but had enough legs, enough proof points to say, wow, this could really, really be something important for patients. And I construed from my stage in career, I was able to take a risk, and also I construed that my experience could be really well suited for that. And here I am, and it's been a great ride already.
Yeah, amazing. Could you talk about the concept of the expanded proteome and what was it about the idea that was incubating at the time that caught your attention?
Yeah, the proteome for all of us has been defined largely by the output of the Human Genome Project and the notion that, okay, let's as a a sort of international effort identify all of the genes and proteins in the human body. And it was a phenomenal undertaking, extraordinarily successful, extraordinarily important. But it was done within the constraints of what was known at the time. And they necessarily had to put some rules in place that defined what would be a protein or a gene. And the output was in the range of 20,000 proteins. And that's been sort of the toolkit we've all been using in drug discovery. Again, very successful. It's somewhere in the range of 400 or 500 proteins from that have been therapeutically intervened with.
Wow.
But we're sort of asymptotically getting to finding all of the tractable targets. And one of the phenomena that I knew very well in GSK, every company has this, is that you see everyone working on the same targets. So if you look at HER2 or GLP-1 or PD-1, there's dozens of companies working on them. And I was always intrigued with finding the novel targets that could lead to first-in-class medicines.
Yeah.
And therefore, this idea was that there's the known proteome. Well, what if there were an expanded proteome beyond those 20,000? And there were some hints at that. For example, people doing mass spec would find spectra that didn't really map to things that were known, and it wasn't clear what to do with them. There were messages that really didn't correspond to, the way we thought. It was like linear, one gene, one RNA, one protein. And, we set up at Flagship, and this is standard for Flagship, to ask a big question that the answer might be, no, that was crazy. Why did you do that? But if the answer is yes, it's a phenomenal breakthrough. And the company was set up about 4 years ago with the notion that there might be additional proteins. And the way we're tackling it is in an unbiased way. So we're asking not, can you find the proteins that are known in cells, but rather, what are all the proteins in a cell? And we use sort of a downstream assay at the level of the ribosome to ask, what are the proteins being translated? And there are tens of thousands of additional proteins beyond the human genome that we now have a very good understanding of, and we're mining those for novel targets.
So where are they? I'm going to ask the simple question that probably a lot of people have. Where are they coming from, if not the 20,000 genes that or so that we discovered in the Human Genome Project?
Yeah, so they don't really defy the rules of biology. They're from open reading frames in DNA and therefore in RNA that were previously unanticipated or unappreciated. And for example, they have alternate start codons, or they're from, they sometimes are smaller. Like one of the important limitations of the Human Genome Project is they set a base of 100 amino acids to define a protein. Right. Many of the ones we're finding are 60, 70, 80, 90 amino acids. And so they were excluded. So these are open reading frames, alternate start sites sometimes, sometimes smaller. And so they don't have all of the rules of proteins. Yeah. But we find them using this ribosomal assay called RiboSeq. And then we confirm their existence with mass spec. And what we found is that, there are thousands of these in cells. They're regulated in dynamic ways. They have drug-like properties or drug target-like properties. They're antigenic. We make antibodies. We express them. It's just a set of proteins that wasn't appreciated to be there.
Yeah. How do you break down what I imagine is a pretty gargantuan problem of the tissue specific and time point specific? Because you have an opportunity here, which is you take that question, which I really like, of what if there are more proteins than we thought, and what if these are doing biologically relevant things for disease that could allow us to intervene? But you have to ask that question on a tissue and time point matrix, right? And quickly you run into this enormous problem of how do we sample this protein space in a way that doesn't break the budget or break any laws of physics of how quickly we can get these things through a machine.
Yeah, exactly. And you can imagine our own investment and other investors are asking that question, like, okay, cool science, but when are you going to have something that's tangible?
Right.
And maybe I just want to emphasize a couple of aspects at a high level, and then I'll come back to some of the specifics. First off, within the company, we have been really clear. We're not here to do human genome 2.0 and sort of define, every parameter of all these tens of thousands of proteins. That's going to take the world's, expertise to do. What we're here is to really have a leading position in identifying these proteins and very rapidly finding those that have features that make them either protein drugs or drug targets. So we've got a very discrete and clear strategy for doing that. First off, and the second macro point I was going to make is, I took the job because I brought to this a notion that we're going to pivot from a pure platform company to a company that has an outstanding platform and a pipeline. And, hopefully we'll have a chance to walk through that, but we've got some really exciting proper drug discovery programs in flight. So how do you go from these thousands and find the ones that, are just sort of poised for drug discovery programs? First step is we created a huge database. We call it our ProFoundry Atlas. It's got 1 trillion data points plus. It's largely data sets that we've amassed from external sources, but also has a very deep data set of our own of these proteins. And because through our assay, the way it works is it's basically we identify as a ribosome is reading the RNA. There's a step, what was an RNase digestion step. And the ribosome is essentially protecting a 30 base pair component of RNA. And we have a mechanism for isolating those pieces and then computationally stitching them together.
Right.
And you find the beginning and the end. And therefore, the RNA that's being translated. You can then therefore know what protein it would have encoded, and what genomic locus it came from. And because you have those 3 pieces of information, we then amassed around that a data set of proteomics. For example, we can search for transmembrane domains or secretion signal peptides or other protein functions. We could make a guess at structure from AlphaFold. And because we know the message, getting to your question, we know every cell that it's expressed in by looking at single cell and bulk data sets.
Right.
So, and then finally, hopefully we'll get to human genetics. We know the genomic locus, and then we could start to, through GWAS and, PheWAS, figure out what diseases it might be associated with.
Yeah, that's interesting. So you can really build on the foundation of all the GWAS work that's been done previously and all the single cell atlas and those kind of programs to sort of stack the layers on the cake.
Exactly. So what we do is now that it took a couple of years to build all this, it's really world class. The RiboSeq, as you'll well know and your listeners will know, is done in labs. But it's very tricky technically. And what we set out to do, and it took a couple of years, was to figure out how to do it in an industrialized way.
Right.
And there are many steps that took like real industrialization, at a platform company level to master. And now we're able to do this on very small amounts of tissue, like 5 milligrams or less. And we studied pushing 400 different human cell types and tissues in disease. So we've got this really deep map of where these things are expressed and how they're regulated. And yeah, on the genetics, it's the coolest thing. I'm just so excited by this. So you're an expert in the field, and you'll be very well aware that most GWAS variants are not coding. And in fact, even the ones that we call non-coding, it's often very difficult to attribute, to really solve what's the variant to gene.
Even one particular element, yeah.
Yeah, and we kind of, we construct stories about maybe it's regulating something in trans that's very far away and there's a looping, et cetera. And, often that might be correct. But what we found is that many of these variants that are non-coding actually are coding variants for proteins that are expressed from open reading frames that were not anticipated to be there.
Right.
So we found many examples where there's a variant associated with the disease. That variant actually is part of an open reading frame that encodes a protein, and the protein has a loss of function mutation predicted by the variant.
Right.
Really cool. So it's a whole new
That's
set
interesting.
of sort of genetically validated potential drug targets.
Yeah, and we'll put a link to this in the show notes, but you all have a nice schematic on this on your website, on profoundtx.com that people can go look at. But what I'm hearing, like listening to this, you have a few different ways you can point this machine. You can go from genetics all the way up through transcriptomics and proteomics to say, can we look at a GWAS hit and explain it now through one of these novel proteins, where before it might have been, we suspect it's regulating a gene in the area, but actually it might not be. That could be a red herring, and it's actually a novel protein that we're looking at. Can you also do almost like a protein association analysis where you're looking at an affected tissue, a tissue from affected individuals, tissue from unaffected individuals, and looking at different protein levels that and make the observation that, hey, we see a greater abundance of this particular protein, and then you could follow that one backwards down the chain, right, and say, is this linked to a genetic
100%.
or other kind of explanation? Is that the right way to think about it?
Exactly. Yeah, there's a couple elements there. One is, aside from the genetics, that's a very powerful approach for identifying novel proteins that might explain either disease process or even response or lack of response to a drug.
Right.
This is a very fruitful conversation we're having with potential pharma partners, where they might have a drug that has important effects on some patients, but lack of response in others. And what we can now have the technology to do is isolate tissue from the relevant, cell type, tissue, organ from those patients with and without response, do the riboseq and identify the proteins that might explain the mechanism for the response versus the lack of response.
Yeah, that's interesting. I can see why you got excited about this when you're hearing about it.
And possibilities are like sort of the questions you could ask are just innumerable. And the way we use the database I described is exactly that, to ask sort of clinically translationally oriented questions. For example, we have a collaboration with Pfizer. It's also with Pioneering Medicine, which is a part of Flagship. And what Pfizer were interested in is which of these novel proteins might regulate obesogenic phenotypes in people. Yeah. So we started that with a list of proteins that have genetic association with obesity. And we haven't disclosed exactly, but we're doing specific pharmacological experiments to identify their mechanism. And, watch this space. This is an area where pharma are interested across multiple different therapeutic areas. And I anticipate we'll be doing additional collaborations like that.
Yeah, that's super. Are there other interesting pipeline examples? Because I was going to go exactly there. You could point this at so many different applications that you talked about, whether it's novel target discovery, repositioning of existing targets, or this response, non-response indicators. How have you thought about how you break down that space, what you do first, or whether you run some experiments with different partners in different places, see what's working? I'm interested in the thought process because you have a breadth versus focus challenge that I'm sure you confront every day.
There's definitely high risk of getting distracted by cool stuff. And, it's one of the things I've brought to the table. One of the reasons I was brought to the company is to have the kind of experience and insight and also the discipline to really prosecute these programs from a drug discovery, industrial point of view, not from like what's the coolest science
Yeah.
point of view. We look for where those two things overlap. One of the areas that we haven't mentioned yet is oncology, because now that we've created this whole compendium of tens of thousands of these proteins and have that database I've described across many dimensions, we were able to ask the following question. Of all of these novel proteins that we've identified, which ones have the following features? They have a transmembrane domain and look like they'd be in the membrane. They have high mRNA expression in cancer, and they have low mRNA expression in healthy tissues. Simple questions that we get asked because we've generated this database. And we got a very long list of those. And we've developed the technologies to now examine which of those is the protein in the membrane, and we know the directionality of it, and it's stable, and we know which cancers. And those have become targets for ADCs or other kind of cancer targeting. And we've got a very interesting portfolio now of novel ADC targets that is very exciting.
Yeah, antibody-drug conjugates for those who aren't as familiar with the acronym, right?
Yep. And of course, they could be targets for other cancer targeting therapeutics, for example, T-cell engagers or even CAR T. It's just for a radio farm.
Yeah.
So they're cancer-specific antigens that are stable and targetable.
How do you break down the causality problem here? One of the things I love about genetics is it gives us some breaks from causality, right? We know it was there, and we don't have this, but you have the challenge here sometimes where you have, is this a cause or a consequence of the difference of phenotype that I'm seeing? And I know you wrestled with this probably for 17 years at GSK.
Yeah, exactly.
Yeah.
Look, it's never easy, and I definitely don't want to convey that this is any easier than the known proteome. The main advantage is it's novel and it's wide open space that we've got a very significant leadership position in. But you still have to do the hard work to figure that out. A couple of aspects, maybe. One is we really try to get the most information out of genetics to really understand which have the most chance of being causative. So there's certainly GWAS, but then we look for replicated GWAS, and we look for with a high p-value. But then we also look for ones that that are predicted to have a loss of function mutation.
Yeah.
And then we also do systematically Mendelian randomization against biomarkers of the disease
Of
for which
relevance.
there's GWAS. And then finally, look for where the protein itself is differentially expressed in the disease with the genetics. So when you have all of those features, those are much more likely to be causative. And then that directs us towards what biology to do. But in the end, you need to do the biology and pharmacology.
How do you think about the strategy of going after, you said a few times in the show how interested you are in going after novel targets. And we've had the discussion with a few different perspectives on this show before of how biotech should think about novel targets, because some people will say novel targets are hard because there's so much to de-risk that large pharma are better to go after it. But also it's where some of the most exciting new opportunities are, right? I'm curious how you think about that.
Look, it's not right or wrong. I mean, companies have their own strategy and their own business model. So there are companies whose strategy is to make a better molecule against a known target. And that's important. You look at HER2, there's like literally dozens of molecules in development with, iterative aspects of the modality. It's important for patients to find, better ways to target existing known targets. But that's just decidedly not our business model. Our business model is to go where people are not. And yeah, you're right. It necessarily adds some complexity. One of the things we think about a lot is when it's a novel target and we're doing screening studies or deeper pharmacology studies in translationally relevant systems, it's easy to get sort of attracted towards when it looks like it's intervening in a known pathway. Okay, so say it regulates TNF and IBD, you're like, okay, I know what to do with that. But that could be a trap because what we don't want to do is find like just a fancy new way of modulating TNF and
Right.
IBD. And sometimes the completely novel mechanism that you never heard of might be the one that's most important. So there's really a dynamic on those two aspects.
Yeah, that is really interesting. What do you do in the lab or computationally to try to navigate that space where you hit on, I'll use an example. We brought up Wave and GSK collaborating on inhibin E as an obesity target. I'm sure that probably was some of the work you were involved in while you were at GSK, but this is an orthogonal pathway to GLP-1s, right? And so the potential there is that you could use them together, maybe a different way to approach it.
Yeah.
That's to your point, more promise there than something that just goes after the same pathway, but in a different way. But how do you chart that scientifically to figure out whether you should continue to chase down that path or whether it's going to lead to a dead end?
Yeah, look, it's different in every case, but I think some of the principles are first off, having people who really understand the disease pathogenesis, having an open mind about the potential for there being completely novel pathways that we don't know about,
Yeah.
which is hard because again, you sort of say, okay, maybe it's like a GLP-1, but that's not the question. The most powerful thing would be something that's completely orthogonal.
Yeah.
And then third principle, designing experiments that take those features into account. So, you know, I, I shouldn't comment on the GSK program, but let's just say in general,
Yeah.
uh, you know, yeah, designing the experiments in a way that take into account like the known mechanism and the potential for an unknown mechanism to be additive or synergistic.
Yeah, it makes total sense. Yeah, I'm curious about you've spent a lot of your time in cardiac, both as a physician scientist, but
Yeah.
also a lot of your work at GSK. Do you see unique opportunities in that world, or are you having to force yourself maybe to take a step back and think more broadly because you have this broad platform? But cardiac is a huge opportunity, I think, to do some interesting science as well.
Yeah, it sure is. So a couple things. I think about cardiovascular, metabolic, even renal as sort of an entity.
Yeah,
And I know a lot of places sort of chop it in different ways or group it in different ways. But those things are so
yeah,
interrelated. And it's played out now with GLP-1s where they've got important activities in heart failure,
yeah.
but likely in coronary disease, certainly in obesity and diabetes. And so I think a lot of the mechanisms are interrelated. Um, it's interesting. When I trained in cardiology a while back now, it kind of was the hot field. And, we're starting to understand thrombosis and lipids and some of the pathogenesis of heart failure, endothelial function. And it was sort of a hot science area.
Yeah.
That cooled off for a while, in part because huge advances were made in those diseases. So the death rate from heart disease and coronary disease and heart failure like dramatically reduced because
Yeah.
of the therapies that came out. But there's sort of a renaissance now of science, particularly in myocardial diseases, in part driven by genetics.
Mhm.
And, again, your listenership will well know that there are now a very important understanding of cardiomyopathy and hypertrophic cardiomyopathy, where it looks like there are molecular subtypes where there's different causative factors for different subtypes. And I think you can anticipate therapeutics coming out of that now. I'm part of a review committee for the British Heart Foundation and just had a chance to look at the results of some of the work being done from their grants in this area. Really cool stuff coming out.
What do I normally when I have somebody as a guest who has spent a lot of time in large pharma, but also in biotech, I ask about the differences. I am curious to hear about that, but I have a slightly different question for
Yeah.
you, just to mix it up for everybody, which is I'm curious what you've learned about what large pharma looks for in a biotech partner, because you have been on the other side of the table a
Yes.
lot. And you know, as lots of our listeners are in biotechs or part of biotechs, and it may not be as apparent if you've never worked at the scale that you have in a large pharma of what actually they're looking for from the kind of partnership that you mentioned that you all have with Pfizer, but more generally, when
Yeah.
biotechs want to partner with large pharma, what are the ingredients for success there from the large pharma's perspective?
Yeah, happy to address that. In fact, I spent a year as the interim head of business development at GSK, and through that experience and just the other
Mm,
roles,
yeah,
did a lot of deals from that side. And it's interesting now. I've had about a year and change experience from the small company looking the other way.
yeah.
Um, Look, it varies, but I think several features. Let's see. Number one, I think the small companies need to realize pharma speaks with a certain language, and if you don't sort of know how to present it in a way that's attractive, that could sort of limit the ability for it to seem attractive.
Yeah.
Then there's, I think,
Is that one, sorry to interrupt, is that one about scale of impact, or what is the
yeah, it's
that
about clarity of
yeah.
what it is, scale of impact, clarity on the stage of,
Yeah.
because some small companies, when they're just starting, they don't have people that have trained in the pharma environment and have never done a drug discovery program. So it's more like a science team.
Yeah. Yeah.
And sometimes they help to really frame it up as, no, it's actually a drug discovery program, and these would be the steps.
Yeah.
And, pharma could usually help articulate that. But sometimes you need to sort of put in front of them in a way that's like, oh, now I see how I could get from point A to point B.
Yeah.
And you actually you have to kind of help the pharma folks frame it up in a way that's going to be attractive to their decision makers
Yeah.
and understand what are the pieces they need for this to be like a tractable business proposition they could get
Right.
to decision makers.
Yeah, that makes sense. So that was one. Any others that spring to mind that are
Yeah,
important?
that's one. And the other direction is, from the pharma side, is to, I'd say, really think through. This is obvious, but it's important. How does really cool, novel, emergent science translate to an actual medicine?
Yeah.
And what are the steps going to be? And what's the probability of each of those steps being successful? And, because the reverse is true. Oftentimes, it's the science team at the large pharma that's evaluating this cool science. But they too might not have the sort of experience and foresight to know, okay, well, how are we going to convert this into a proper drug discovery program that leads to a medicine, while we're all still here?
Yeah.
And, that's the part that pharma has to get really clear.
With Profound in mind, on that second point, one of the things that I was curious about was how you think about different therapeutic modalities, because it's
Yeah,
another
good question.
focus versus breadth question. You
Yeah.
have, you can go for, you discover one of these novel targets, and you've got a huge toolbox of how you actually go about treating it, right?
Terrific question. The way we're doing this is decidedly not to iterate on the modality. So we're looking for targets that could be addressed by existing modalities,
Yeah.
standard modalities. That way, you're not dealing with two experiments. We've got a novel target and a novel modality, which just increases risk.
Yeah.
And by focusing on standard modalities, it also is more efficient and reduces time. So we're focusing, for starters, on programs either where the genetics tells us the directionality, where the protein itself would be the therapeutic,
Yeah.
like a glucagon or an insulin or a GLP-1, or where we would modulate the protein with an antibody.
Yeah.
We're not having small molecule programs for several reasons. It's very expensive to stand up the organization. And also a lot of the proteins we're working on are smaller, and they don't have sort of standard,
Right.
pockets and domains for small molecules. So yeah, we're focusing on standard modalities.
Yeah, yeah, that makes sense. I was curious about, I mean, that one of the things that's tantalizing and interesting about this is, to your point about you may discover a protein that is linked to protection and some kind of general principle, right?
Yeah.
How much are you thinking about proteins. Like, I'm just curious about the universe of cases where protein overexpression is the problem, and so you're looking to modulate
Yeah.
down versus we have these interesting examples in there's a number of them, but in genetics where presence of the protein or some version of the protein is actually protective. And this really can crack open a whole new field sometimes, right? Where you find a very small number of people who have a very
Yeah.
protective thing, but that becomes a window into if we can produce this and give it to everybody, then it's the key. Is which of those, if you were to weight between those two, do you have a sense of where the most interest and excitement is going to be?
Yeah, I'm not sure it's I'm not sure weighting the approaches is the right way because I think this is very specific to the target and the disease
Yeah.
and circumstance because there's a couple of other layers there, right? There's like, is it possible to deliver enough? Like, what's going to be the requirement from a sort of a PK and drug substance point of view? What's going to be the delivery route? What cells does it need to get to? I could have to inject it, or could you give it long-acting systemic? There's just like a lot of layers there, but clarity on this question is absolutely essential at the time you start the program.
Yeah.
Because especially for a small company like ours, where you can get waylaid by this like really cool, as I was alluding to before, cool biology. But if you think through all the steps, and I'm going to have to make, gram quantities, per
Yes.
patient every day, that has to be intrathecal. Like, all right, that's not the first place to start for a company like ours.
Yeah. I understand that you have a at least from my where I sit, unique but really refreshing way of how you think about driving priorities and the big picture within the company, where you ask and encourage everyone to ground the science and the work they're doing and how is this actually going to help the patients that we're focused on. I'd love to hear a little bit about where that came from and how you apply it, because it's easy to get distant from that and impact that you're trying to have when you're doing the science every day. And I'd love to hear a little bit more about that.
Well, thanks for asking. I'm very passionate about this. And if any of my GSK colleagues from the old days will remember, I used to do a lot of like walking around the lab and just sort of asking scientists. They would think I was going to ask them, well, what experiment are you doing? And, how to
Right.
tell me about that. But I would ask, well, hey, tell me what medicine are you making? And for some of the, early career scientists they thought I was pipetting the B cells to do whatever. I'm like, no, 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 you're making a lupus drug. So like,
Yeah.
I need you to be able to explain how does your pipetting today relate to that? But it's really important for efficiency and productivity, because if you're just distracted by things that in the end aren't going to be able to be clinically applicable, that's just like wasted effort. And I just feel passionate about it because again, as having been a physician, I just sort of saw what the patient needs are. And it's important to do basic science. I don't want folks to sort of forget that. And, like supporting NIH, for example, to do like real basic science that does not necessarily have a translational endpoint is extremely important for society. But when you're a company that's there to make medicines, that's not your job. Your job is to make medicines.
Yeah, and I think it's a really nice way of making sure that wherever you are in the organization, you can tie the work that you do back to that ultimate purpose, right? That, like, exactly like you said, that's the ultimate purpose of a company like GSK that's making medicines. If you're in an academic environment, your ultimate purpose might be find the truth, find new science, and you could apply this same principle there, right? Of what am I doing today that actually ties back to this mission, which is find the
Yeah,
truth of the
yeah.
universe, right?
But for clarity and to remember what hat I'm wearing, it was definitely important when I was at GSK, but it's
Yeah.
also very important for ProFound and for our
Yes.
company at this very early stage. Like it's called ProFound Therapeutics for a reason. And, I tell the team all the time, I think I mentioned this before, we're not human genome 2.0. We're a therapeutics company that's using a science and a platform and a technology in a new area to find new targets, start programs, and make the medicines.
Yeah. Amazing. As we wrap up here, what should people expect from you all over the next couple of years? Do you have a sense of when you're going to start moving some of these into first in human studies? How many, with what kind of pace? Don't answer anything that you're not allowed to answer or can't
Yeah, okay.
don't want to be able to walk back in the future. But I'm just curious to get a sense of where you all are.
Yeah, look, it's an exciting, burgeoning field. we have the luxury of now being in a very strong position of having identified probably the vast majority of these proteins in a highly competitive way. And we've got them in a context with this data set where we could sort of pick the cream of the crop of drug targets. We've started drug discovery programs across oncology, immunology, and metabolism. And I learned long ago not to give timelines for specific endpoints. But look, these are real drug targets that will have timelines similar to known targets. It's just we found them first, and we're going to stay focused and move aggressively on them. And I think you'll also see us continue to expand our partnerships with other pharmaceutical companies over the months and years. Because this is a field, obviously, we can't build a huge company rapidly to do all of the good work, and we'll be doing a lot of it as pharma partnerships as well.
Yeah, absolutely. I mean, you joined the company in 2023, right? And on LinkedIn, you're about 40 people. Probably a lot of those are advisors, investors. So it's amazing what you can accomplish with a small, smart, dedicated team in a short amount of time.
And a focused team. Yeah, that's right. And passionate about making medicines.
Absolutely. Well, John, thank you. This was great. If people want to keep track of you and your work, we'll make sure that we put the website, profoundtx.com. Is there anything else in particular? Are you hiring certain roles, looking for other kinds of partners that you want people to reach out specifically on?
We're definitely looking for people passionate about applying this cool area of emerging science to making medicines and doing something important for society and science. So give me a call.
Amazing. All right. Thank you. And thanks everyone for listening, and we'll see you next time.
Thank you, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.